somatic embryos developing into a new plant. So, during this development, roots and shoots are formed. So, the root and shoot formation is depending on the auxin and cytokinin concentration ratio. And here organogenesis, different organs are organogenesis. Here root formation, called as rhizogenesis. Here roots are formed. And shoot formation. Shoot formation is called as calogenesis. So, root and shoot are formed. After the shoot formation, that producing the leaves, fruits, so all the parts also. So, this root and shoot formation is called as organogenesis. The organogenesis, it is depending on the auxin and cytokinin concentration. So, generally auxins induce the root formation, and cytokinins induce the shoot formation. Root formation is induced by auxins, phytohormones. Shoot formation is induced by cytokinins. Here shoot is formed. So, the shoot is formed and the shoot is developing into, the shoot is producing the leaves, flowers, fruits and seeds also. Within the test tube also we can develop the plant. When all the food materials are provided to the tissue, so the tissue is developing into a new plant. So, the tissue is developing into, or one cell also, one living cell or some tissue is developing into callus. Callus is developing into embryoids or somatic embryos, embryo-like structures. So, the embryo-like structures are developing into a root system and shoot system. So, the complete here one plant is produced from the single cell or from group of cells also. So, in the tissue culture technique, we can produce a number of plants within a short period, within a short place. So, that technique is called as micropropagation. So, by using this technique, tissue culture technique, we can produce number of plants. So, number of plants. So, in large quantity, here we can produce the plants. It is called as micropropagation. Within a limited space and within a short period, we can produce here some thousands of plants also. So, it is called as micropropagation. By natural or traditional methods or conventional methods, here some plants, the breeding is generally after the breeding, or the plants are producing the seeds develop into plants, but the plant production is it is taking so much time. So, here by micropropagation technique, by using micropropagation technique, we are producing large quantity of plants. So, we are a number of plants, we can produce a number of plants within a short period and a short space also. So, it is called as micropropagation. So, by this micropropagation technique, here number of commercial plants, commercially important plants, economically important plants are produced in large quantity. So, it is about micropropagation. And next, so protoplast fusion. So, protoplast fusion is nothing but here the protoplast, both protoplasts. So, here if two plants are having desirable qualities, the desirable qualities or desirable characters are present in two different plants. So, the two desirable characters are incorporated into the single variety or single plant. So, it is possible by protoplast fusion. Here by using traditional or conventional techniques, crossing the two plants, it is here are some complex process, but here the protoplast fusion, the union of protoplast. So what is protoplast? So within the cell, within the plant cell, here one cell wall, outer layer, outer boundary is cell wall, and it is the plasma membrane. It is nucleus. So if we remove the cell wall, if the cell wall is removed from the plant cell, the remaining material is called as protoplast. So completely the remaining material is called as protoplast. So the cell wall is, when the cell wall is removed from the plant cell, the remaining material is called as protoplast. So one living membrane is there, and inside protoplasm is present. So the total inner contents called as total, including plasma membrane and protoplasm, it is called as protoplast. So the protoplasts from two different plants, they are fused, they are combined. So the protoplast fusion, here protoplast is the protoplast is nothing but a protoplasm, plasma membrane, plus protoplasm. So it is plasma membrane. Inner contents are called as protoplasm. It is a protoplast. So one protoplast, this protoplast is from one cell. The protoplast is from one desirable plant, and this protoplast is from another plant. So two protoplasts from different plants, they are taken, and the cell walls are digested. Cell wall components are generally cellulose and pectins. The cellulose and pectins are digested by cellulase enzyme and pectin enzymes. Pectinase enzymes. Cellulose and pectins are digested by cellulose and pectins are digested by cellulase and pectase enzymes. Cellulose is digested by cellulase. Pectins are digested by pectase enzymes. So the cell wall components are digested by cellulase and pectase enzymes. After the digestion of cell walls, the remaining components, the remaining material is called as protoplast. The protoplast is from one cell or one plant, and this is from one other plant. So both are having desirable qualities. These two are combined. So when the two are combined, it is called as when the two protoplasts are combined or fusion. So the fusion is by here electrofusion, sometimes chemofusion. Here polyethylene glycol, by using polyethylene glycol, or it is one chemical, by using chemicals, by using here chemofusion, by using chemofusion or by using electrofusion. Here we can combine the two protoplasts. The two protoplasts are fused. So after the fusion of protoplasts, sometimes two nuclei are present. Sometimes the two nuclei are also fused. So two nuclei are fused. Only single nucleus is present. So sometimes two nuclei are present, and the when two nuclei are also fused, only one single nucleus is present. So here, if this protoplast from tomato, if this protoplast from potato, so in tomato the fruits are useful parts, and here the potato, in the potato the tubers, if the two characters are present in the same plant, so that is a very beneficial character. Commercially it is beneficial. So that's why here tomato and potato protoplasts are fused with the help of here polyethylene glycol. After the fusion, here the single protoplast, fused protoplast. So it is having one nucleus or two nuclei also present. Here the when the two nuclei are fused, it is called as here hybrid protoplast. The hybrid protoplast is cultured on the tissue culture nutrient medium. So after this fusion, so this hybrid protoplast, it is hybrid protoplast. The hybrid protoplast is cultured on the medium, on the nutrient medium. It is when it is cultured, that develops into new plant or hybrid variety, and it is having all the characters, all the two plant characteristic features. So the produced plant is called as here hybrid plant. Tomato, potato, protoplasts are fused, and hybrid protoplast, when it is cultured on the medium, that develops into a plant, that is hybrid plant, having called as a pomato. The pomato, it is unfortunately it is not having the all the characteristics. So here only a few characteristics are present of these two plants. So pomato is it was not successful commercially. So pomato, but here we can fuse the two protoplasts from desirable plants, and the hybrid protoplast can be cultured in the tissue culture medium, and that develops into a new plant. It is a protoplast fusion. By using protoplast fusion, we can fuse the two protoplasts of desired plants. By using tissue culture technique, we can produce the plants, required plants in large quantities within a short period, in short space, limited space. It is micropropagation. And we can produce haploid plants. Haploid plants are, what is haploidy? Ploidy, the number of sets. If one cell is having only one set of chromosome, so that is haploid. If two sets are present, that is diploid. If three sets are present, that is triploid. So the ploid is nothing but haploid. Haploid is nothing but the single set of chromosome. If one genome, here human cell, here human cell is having 46 chromosomes. Here the 46 chromosomes are arranged in 23 pairs. Here 23 and 23. So 23 is one set coming from mother, 23 is other set coming from father. So two sets during the sexual reproduction. So one set is from egg cell, one set is from sperm cell, or male and female gametes. So two sets are present. So one set is called as one genome. So it is one set. If only one set is present, that is a haploid cell. If two sets are present, here diploid cell. So the ploid is nothing but the number of sets of chromosomes. Here by using anther culture, we can produce the haploid plants. Haploid plants can be produced by using anther culture. So anther is the part of stamen. The stamen is having here the pollen grains. In the stamen, it is a filament and it is anther. The anther is generally producing the pollen grains. The pollen grains are produced from the pollen mother cells. Pollen mother cell is dividing meiotically to produce 4 haploid pollen grains. So when the anther or inner tissue, when it is cultured on the medium, so the pollen mother cells are dividing meiotically and producing the pollen grains. The pollen grains are developing into, each and every pollen grain is developing into a plant, a new plant. So the plant is, here pollen grains are generally haploid. So pollen mother cell is diploid but when it divides meiotically, so haploid pollen grains are produced. So the haploid pollen grains develop into new plants. So here haploid plants can be produced by anther culture. When the anther is cultured on the medium, the anther develops into haploid plant. These haploid plants are here used in the other hybridization or other breeding techniques. So here some plants are triploid plants. When the haploid plant and the diploid plant, when the two are here crossed, a triploid plant is produced. Triploid plant. So in the breeding technique, generally haploid plants, wherever they are necessary, wherever they are important, so these haploid plants are used. So when the haploid plants are here crossed with the diploid plants, here triploid plant is produced. So one example is here Citrullus vulgaris. Citrullus vulgaris. Here seedless watermelon. So Citrullus vulgaris is one triploid plant that is produced by this method. Okay, here by using anther culture we can produce haploid plants. Haploid plants are here used in the breeding techniques, plant breeding techniques. So it was anther culture was firstly developed by Guha and Maheshwari. In Datura inaxia. In Datura inaxia plant, the anthers are cultured on the medium and haploid plants are produced. First haploid plants were developed by these scientists Guha and Maheshwari. And embryo, here embryo culture also, embryo culture or other parts also we can culture or we can develop into new plants. So sometimes the embryos are immature. After the sexual reproduction, the zygote is developing. Zygote develops into embryo. The embryo cannot develop into new plant sometimes by any reason, either genetically or sometimes it is by environmentally. So here immature embryos. Some embryos are immature or they cannot develop properly. So this type of embryos are taken from the seeds, and these embryos are when the embryos are cultured on the medium, here the embryo develops into new plant. So immature embryo, for example, it is immature embryo. This immature embryo is cultured on the medium, so it is developing into new plant. So naturally it cannot develop into new plant, but the immature embryos are taken from the seeds. So it is a seed. In the seed one embryo is there. So this embryo is taken out and that is cultured on the medium and that develops into new plant. So like this here these are some uses micropropagation and anther culture and immature culture. So here by using tissue culture technique we can produce the plants in large quantity. So commercially or commercially important plants are cultured in large quantities within a short period, within a limited space. And haploid plants are also. Here in the plant breeding technique, some haploid plants are used. So the haploid plants are developed by this technique, anther culture. Sometimes immature embryos also can be developed in the tissue culture technique.